we're ready for questions, but are there any comments from the end?
Thank you, Daryl. So I think you've seen that the name of the game here is all about identifying, quantifying your sources of variability. That's what this is all about. And in order to do that, you need to look at your relevant data. And most of the time, that's going to require data integration with a lot of curation attached to that. The comment that was made by Sandy about adherence is so critical, because you have to get the dose right, and then patients need to take the drug. Other than that, it's not going to work. The fact that EMA is currently at the public review process for a qualification opinion for a digital biomarker to measure adherence together with aripiprazole, speaking about schizophrenia, is I think a testament to the what we're going to see in terms of increasing use of digital biomarkers, and how we can use that data to inform the models. And then naturally, if you want to build a clinical trial simulation tool like the one we built with CMD that Brian presented on, you're going to have to account for the placebo effect. And I'm really happy to see that, the placebo effect function in our model is purely empirical. It's great to see that people like Ariana have not only done the empirical modeling part, but are starting to delve into the more mechanistic features of how you can actually account for that effect by using neuroimaging. So the more we get to integrating those data, we can get into what Sandy showed about mechanistic models that account for the pathophysiology when you know the pathophysiology in HIV, or try to explain the pathophysiology in fields where we don't know so much about that, like CNS indications. And to wrap it all up, we're going to have the regulators at the table to provide some level of endorsement for the models so that they become platforms that all the sponsors can use and they themselves can use, so that when you're having a regulatory conversation for an NDA, everybody is speaking the same background quantitative language. That's going to expedite things, and that's going to do away with a lot of the guesswork that is currently so prevalent.
Thank you. That leads into Brian Corrigan. I was going to ask really almost what you alluded to. When you're thinking about a model for a disease for which you have an understanding of its mechanism as compared to one in which you don't, do you have thoughts about, kind of how you would evolve that model and how you'd really test its utility, particularly in the context of interventions that may or may not be effective?
So I think when you have an understanding of the underlying, hopefully in most cases these days we have some understanding, and I think the other component to think about is where it is in the drug development is it early development, later development, and what questions you're trying to answer with the model. So for example, usually we will incorporate the real mechanistic understanding into something like a quantitative systems pharmacology model. And that may be used very early on in either in translation from animals to humans, or sometimes for understanding of safety in later stages of the program. We might use much more of an exposure response type of approach for sort of the midlife of a compound. So in that case, you may have some of the things may play a role. We might use physiologic approaches to understand PK, for example, to explain the exposure, and then relate it to response. In later stages, we might use the more empiric approaches like we've shown here, and or even the literature meta-analysis or the network meta-analysis types of approaches to inform us about the competitive landscape, or to help support, for example, health economics type of approach or question. So I think it's really more dependent on what question you're trying to answer.
Thanks.
Maybe I think Sandy had alluded to it also in her talk.
Well, I think that actually is the most important part. It's not about building a model looking for a question. It really is about clarifying the question, what are the data that are available, and using those. And we do see the more quantitative and systems pharmacology models available early on as to help us with hypothesis generation. But as we hone in on the more critical aspects of that, then you can kind of go to the middle out and lift up the pieces, because the more mechanistic we can become, the more likely they're going to be predictive. Because if our goal is to get away from getting the dose, one dose for everybody, and really moving towards understanding the right dose, then we have to move to a more predictive and more mechanistic approach. But it's challenging.
Questions from the floor?
I wondered if I could ask a people question that you raised, being the crucial question. The Alzheimer's CAMD work that you built, trial simulation and design tool. I'm interested in who the target user is. And the reason I ask is that, you know, if it's going to be the physician, and it may not be at Merck or at Pfizer, but in some places it's going to be the clinician who kind of decides and drives the trial design more so than the modeler. But if the model is something that is more suitable to be used by a modeler, you know, if it's an R, if it really needs expertise or etc. I'm curious who the target audience is for your model and the degree to which you've been seeing it used.
So I can address the target audience. Maybe Klaus can address how much it's been used. So in our case, this particular model, this particular tool is designed for people who will be supporting, asking questions about trial design. I think you're right to say that the clinicians on most drug development teams are in the decision-making role. They are not the ones that are doing the trial simulations, with probably very rare exceptions. So this particular tool and the technical components, it's designed, it's a nonlinear mixed effects type of model, written into our code and as a series of functions to allow simulations. It takes time. And to do simulations appropriately takes time. So in this case, it may take hours to do. So it's designed, this particular tool is designed for statisticians predominantly, or statisticians and modelers. And I think that was a key learning also. So, you know, this was built, you know, when this came out a few years ago. Mm-hmm. Now it's very easy, for example, to take our code and make it into an application. It's really easy. I mean, there's packages in R called like Shiny, for example, that allow you to do it in a matter of hours. And so you can put this into the hands of clinicians as well, if they don't have the statisticians. Although sometimes understanding the operating characteristics of a design, it's probably useful to have a statistician around. Correct me if I'm wrong.
It's always useful.
I stand corrected. It's always useful. Yes. So, and then Klaus, maybe you want to answer the question about the usage?
Yeah, sure. So we've been, we are working with FDA and EMA to actually get that formally quantified in a way that is shareable. We know that since the model was endorsed in the summer of 2013, every NDA that has come to the agencies regarding mild to moderate Alzheimer's disease has used, if not the whole shebang with, you know, the disease progression, drug effects, placebo effect, dropouts, at least a portion of the entire tool. So both the sponsors and the regulators are happy with that, but we're trying to formalize how we can actually share that information without compromising any of the ongoing programs, et cetera. By nature, these kinds of tools have to be evolving creatures, because there's always going to be, I mean, all models are wrong, some are useful, we want to make them useful. So we're going to have to feed in additional data as those data become available. Biomarkers is the next target. Moving into the MCI space is the next target. And the other target that we have in mind, when we actually had a Corrigan committee meeting last week about this, is to invest in developing those kinds of interfaces for the less modeling prone type of person to be actually exposed to those tools and be able to, if not use the whole universe of potential applications, at least get a feel for what it looks like and how that can help answer specific questions about trial design.
Just say kind of tangentially, I alluded to at the end the kind of trial that we're developing that's more of a global trial. And the one thing I found that's really interesting about that whole process has been, it's what Ann Barker called a crowdsourced trial. And it really requires a lot more and different kind of interactions among people, normally, and it's really fascinating to see. Like, it used to be, the neuro-oncologist would come up with a trial, and like they would say, this is what we want, how many patients we want to enroll, and all these kind of things. It'd be very simple, but this is a lot, and it's fun in a way where you actually have to examine your assumptions and say, what are we actually trying to get out of this? What information are we trying to generate? And then there has to be a lot of really good teamwork involved in kind of going back and forth to kind of come up with the design elements. And it's a lot of like siloed knowledge that it's starting to bleed a little bit across borders, but it really requires a whole different kind of a social environment than I think was before in clinical trial development.
It's very cool.
Yeah, one of the things, when we think of modeling, it's not a quantitative scientist that's doing this. This belongs to the whole team. It belongs to the experimentalists, the clinicians, as well as the modeler. And it's about putting them together. And where we've seen the greatest value is really looking at those assumptions and how much receptor occupancy do you need to see an effect, and what if we're wrong, and where does the trial begin to break with that. But if we just go away and build a model and then come back, then if it gives them the answer they like, it's pretty good. And if it doesn't, then it's less likely to be good. And but if they've built it with us and they understand it, and I think some of the new tools like Shiny and some of that's really, where they can interrogate the model. It's not just take our word for it. It'll help.
Yeah, but I just want to be careful. And again, we want to leave the right tone. All models are wrong, but so we sit here as quantitative scientists and sort of lay out our assumptions for the world to see. But what I tried to present in that slide is that we generate a lot of data. Most of our organizations don't really have a knowledge management structure to accommodate all those types of data. All those heuristics and biases that were sort of outlined by Daniel Kahneman which he won the Nobel laureate for in 2002, right? They're all at play. So, you're not having a model is a model in and of itself. And it's basically just leaving it open to the heuristics and biases that most drug development teams or most decision makers have for whatever, right? At least what we can do is we can explicitly put them out for you to look at, observe, touch, feel, poke. The same isn't true if you don't have one. And in a consortia, you can have more people touch and feel and poke those assumptions, which should give you, hopefully, if you have a bunch of quantitative people, better answers in the long run. But it's not models or something or the other answer. Everything is actually, the other part is a model, it's just not explicit.
Other questions from the floor? Comments?
I guess the other thing that I would add to Brian's comment is yeah, I mean, if you have a model, you understand your assumptions, you understand your gaps. That's a quantitative way to inform the gaps. So you can circle back to the experimentalists in the non-clinical space or even in the clinical space and say, we don't understand this. There's a gap. Let's try to find a way to fill that in. So that's in itself an advantage, because then you can prioritize which gaps should you fill to actually be able to inform the model to the next stage. And then the other part is, sure, you can go at it with a crystal ball and ask a bunch of experts and try to hope for the best, or you could base it on, however limited, but at least a reliable quantitative estimation of what you have and what you can do to predict what's going to happen. So it's, yeah, It's a worthwhile investment, but of course, we're all biased here because we like models.
I was thinking, spoken like a modeler. So,
You really do have to get into the weeds too, though. I mean, like for clinicians, and I'm a clinician. You know, there was a model that we were looking at one time that was based. It was an adaptive randomization model that was based on based on whatever it would randomize based on whatever would provide the most information. So you know, it was designing the best. The next patient would be randomized to whatever arm would provide the best information. But then you kind of look at it and you're like, well, if an arm is doing really well, and it knows that, then it stops randomizing people to it. And so you're kind of looking at that and say, well, you know, I don't think that makes a lot of sense in terms of like a clinical trial. Like a patient would want to be enrolled on an arm that was actually doing well. So, you know, there can be like small assumptions that are made within the design that have really big implications, and you really kind of got to get into the weeds. It's a lot more fun and it's a lot more interesting. But I think that there's a lot more abilities for kind of the assumptions to translate into kind of things that you might not have expected.
Great. Alistair?
Models, what responsibility do you see yourselves all having to make clear the parameters that have gone into the models? So later on, we're going to see feedback from these models. And if it's a black box that we're working with, how are we going to interpret that? And how do we put that across in publications or even without publications?
I'll start. I imagine we all have an opinion on that. I actually think that's one of the most important questions, and probably what you're asking, it's what's model qualification. And I think it starts with being transparent about the data that you used, because the model is only as good as the data that you're using, and you have to know what those data are in order to know how you can use it for other questions. So it's being transparent about that. It's being transparent about model structure and showing where the model, being able to come up with tools to show where aspects are fitting well and where they're not. So for example, an early model that we had developed on looking at the ability of our drug to prevent stroke or cause bleeding. If we were using, it was based on comparator data as well as our own. We were doing really well at our ability to predict whether we were going to prevent stroke. But if you looked at our model for preventing bleeding, it became quite poor. And we had to show that, and primarily because there was a paucity of data. You didn't want to put anybody up at a risk of causing a hemorrhagic stroke. But we had to be very clear that you could take this with some confidence, and this part you had to take with care. But then you could run the simulations and look, well, then what's the probability that the dose would do what we think it is? And in this case, it led to a termination of the compound. But it allows us to interrogate. And I think that's why it's really important. It's not a point estimate from the parameter. It's really that range and what's the probability with it. So I do think understanding how the models have been qualified, how do you use them, have they been pre-specified, that's all aspects of sort of best practices. That's important for folks to understand, just like you would read a clinical paper.
Remember what patients were included in the model to make the model, because they might not extrapolate to other age groups or whatever.
That's exactly right. What are your data? What's the range? So if you're trying to look at a pediatric patient population and your youngest patient's 24, it's probably not very predictive. And that's exactly right. So it's truly understanding what we find, for example, on the literature data where we're pulling that in. We sit with the clinical team and we go through paper after paper. Are these endpoints the same? What do you have to find? And I think it was mentioned earlier this morning, if there's a biomarker where the assay is the same, we've had to go back and find factors and ways to be able to leverage those biomarkers in our hands. So those are all important questions that the team, and when we call a modeling team, it's again, I keep coming back to experimentalists, clinicians, all together, asking these questions as you go. And if we're going to then lift and shift these models and use them for other cases, we have to be transparent about how we got there.
Well, I think a part of that question, at least to me, is that there are frankly a variety of vendors out there who use a variety of different kinds of models, and they view really the value that they hold is the black box, and those models are not transparent. I say I personally have strong feelings about that these models should all be in the public domain, and the data supporting them should also be in the public domain. But I'd be happy to put that on the table for discussion, because perhaps we're not all in agreement about that.
I actually could not agree more with that statement, and that's kind of the whole philosophy behind the Alzheimer's clinical trial simulation tool that we put together. It's not, the data are publicly available, the code line and the supporting files are all publicly available, but on top of that, well, we issued publications and whatnot talking about the limitations and the data behind it and whatnot, and the parameters that went into the model structure. But the icing on the cake is actually getting FDA and EMA to review it, review the data, review the code. They even helped us optimize the code to a certain degree. So in the endorsement letters, qualification for EMA, fit for purpose for FDA, all the limitations and all the advantages and the clear context of use is described with a few examples. So if we get to that level of a conversation where we have the regulators on board with reviewing these kinds of tools, then we'd be in a place where, as I said, everyone will be speaking the same background quantitative language.
And I grew up on a farm in Canada, so by definition, I'm a pragmatist, right? And so, new modelers in my group that come in, the first place they go, the first course that they take is Toastmasters. So that, that they have some self-awareness of how they're talking to people who may not necessarily be as quantitative. They typically are very well trained in the quantitative sciences. In explaining the quantitative sciences, they're not. Coming back to the question about should the models be explicit, I couldn't agree more. And it's the only way that we can test the assumptions around and sort of test whether they're waterproof, and whether the clinicians and the statisticians and the other parts of the team can really be comfortable with the decisions that are being made from those models.
One of the things as we think going forward, our models are as good as the data that we can acquire. And right now, many, even in a classic population PK/PD analysis, how many samples can you get from any one patient? We're lucky if we get 3 to 5 over a several-year period. And so we're leaving so much information on the table. So I think one of the things I'd love to see this group think about is, are there new trial designs? Are there new ways to acquire data? How do we really think about changing those designs, which will then allow us to do a lot more with that data towards getting to that predictive understanding?
Yeah, it's a question over here. Comment?
So it feels a little bit like one of the ways you're framing the potential uncertainties or managing the risk around imperfections in the model is that there'll still be some definitive trial afterwards in real patients. Yet we've also had a conversation today about how those trials don't actually produce perfect information. And in fact, there was even a discussion earlier about there could be a situation where understanding the quality of an information around a phase 3 could result in approval if we knew that there would be something happening afterwards in phase 4, which presumes that we're spanning across the space of the different experiments. And the best way to span across the space of the different experiments is probably some integrated model. So could you comment on how you could see us getting to a point where the best and final sort of repository of the information is the model, that there's no longer saying, don't worry, we'll check against the data later. So the model would be last
Yeah,
word.
that's actually the way, I don't like, actually don't like talking about models that much, but knowledge management is something that's very near. If I could go back to university, can't afford it. I have a student in college, so I can't afford it. But if I could, that's what I would study, because it's really the question of, you've seen all the data that's being generated. We best have a plan for how we're going to, store it in a meaningful manner that we can reuse, and then integrate it so that it actually becomes knowledge. And so if you frame the question as a knowledge management question, how are we going to store this data, how are we going to integrate it, you kind of sort of only come to one conclusion. There's going to have to be some sort of integrated approach to make sense of this. And that's really what a knowledge model is. It's just a mathematical description, and that's probably all that we can really have of all the information that we have within a program, in some cases maybe beyond the program, across a mechanism or across a disease area. That's all it is, right? And it's mathematical, so we try to be explicit in the assumptions. But that's the way to frame it, not as a model and the assumptions are wrong.
Well, and I think kind of part of what you're suggesting, kind of what's the level of comfort in the outcome from a model-based exercise as compared to really a data-driven exercise? How do we balance those two things? Is there a way that we can move more toward outcomes based on more model-based exercise in order to decrease needs for clinical trial burden and the rest? I think that's a very interesting question. I'd be interested in people's comments.
Well, and to that end, I believe at the American Society of Clinical Pharmacology and Therapeutics pre-conference sponsored, or one of the co-chairs was, I think, Vikram Sinha from FDA. The conversation really was around, are there ways, for example, when you have a very good understanding within a program, as you suggest, that can sort of not be confirmatory evidence, but supportive evidence in cases, for example, maybe for a special population where you may be suggesting a different dose and you can't necessarily study them, or it's difficult, or there's not enough patients. Or even perhaps at some point, that you have the one confirmatory trial and supportive evidence approach. So that's being discussed.
And I think the next step for us is to do away with the concept of phase 1, 2, 3, 4, and think of it as seamless. And what do we know? What are those knowledge gaps? How do we inform it? And we are seeing the quantitative approaches accepted through the regulatory process, especially as we're getting more mechanistic. But I think it's being very clear on what data we have and how do we go forward with it, and what are those knowledge gaps.
I'd take the opposite approach. I kind of am a little bit comforted by the fact that there's a confirmatory kind of classic randomized trial a lot of times. Where, I'm not worried about the transparency in the models. A lot of times I'm just worried about kind of the communication between modelers and clinicians and everybody, and actually being able to understand the assumptions that are made. And so, I think the current state of play, at least in neuro-oncology, had been to run a phase 3 trial with maybe single-arm phase 2 data or phase 3 data from another tumor type. And I think generating information to better prioritize those phase 3 hypotheses is a good step in the right direction without going all the way to kind of the seamless through phase 3. I mean, I'd certainly love to get there, but you do make a lot of assumptions in certain cases, and you can't. I would say that when we're modeling different kinds of trials, really coming up with all the different things that could happen in terms of the biomarker frequency, the biomarker response, the response rate in one, are the auxiliary endpoint relationships with overall survival the same in all biomarker categories and all therapeutic categories, and what if we were wrong? I mean, there's just so many things that you could come up with that you might not get right, that having a kind of confirmatory phase 3 that's a randomized trial, as you showed it, I think is at least not so bad.
And I don't think that's what Vikram was advocating, at least one full randomized trial. But when I talk about doing away with we're here to phase 1 and then we're moving to phase 2 and 3, it's like treating those like they're individual. We threw away that piece of information and now we're into this next one. The molecule and the model are learning together as we go through each step. And I think it was Brian, I don't remember, one of you guys said, if as we understand, there's an assumption here that's really critical, that the whole, it breaks if this is not met, then that may be unacceptable, and that's where you go get the data. So that's where we've seen the greatest value, is understanding where those gaps are.
I think it's yeah, I agree. I mean, I totally agree with the kind of information flow through the continuum. I hope I made that clear before, and particularly when it comes to biomarkers, because I think we very much distill down information from, we have these kind of basket trials which have no real rationale for some disease types other than like some preclinical study in a different tumor model or something like that. So having biomarker information generated throughout the course of the spectrum, I totally agree.
And it kind of gone full circle, but I think it comes back to the better one understands the mechanism of disease and the target that they're trying to achieve within that mechanism, perhaps the more confidence one should have in the model itself. My usual anecdote here is that, we can model the acid peptic hypothesis of gastric ulcer disease and probably come up with a great model, but that's not the disease. Yeah.
So I understand, and maybe the hour is late, but I'll just give one riposte. So part of it might be that we get great comfort around the word confirmatory, because we've actually narrowed down to this little single moment of sort of drug approval, which is artificially small, because it's not really the end of knowledge even in that disease, let alone the knowledge in the drug. And so you could do a thought experiment where you could imagine you were regulating something else, like you were regulating timing of a scarce resource like liver transplant. And there's no trial to test the optimal timing for liver transplant, because optimal timing is almost infinite. You would take people with end-stage liver disease and try to transplant them at any time over a 5, 10, or 15-year period of end-stage liver disease, and you didn't even know when the end-stage liver disease began. And so under those sorts of problems, they're really only informed by models. The final and most complete evidence on optimal timing is modeling the size of the transplant centers, the projection of the disease, estimating the disease in the absence of transplant, and so forth. So if we substituted the notion of a phase 3 approval as a thought experiment, imagine that didn't happen, and thought about sort of drug approval as more in a continuum for which there is no single confirmatory trial, then we might actually elevate the level of these sort of integrated models.
On that thought, I believe we're at 5 o'clock. I'm seeing shaking heads here in front. And so thank you all for your attention. I don't know if there's any closing comment or not, but thank you for a good day.
Well, Stephen and I are given the unenviable job of, in 5 minutes, giving a wrap-up of session 1 and session 2 after 9 hours of this, which really is unenviable. So what did I take from session 1? I think I heard the message that there were real consequences in moving from a data-poor, opinion-rich environment to a data-rich, opinion-rich environment. And that part of that was that we had to start to develop systems to take account of all of the data to which we have access to. And we had to start to think about how we do that, which is a non-trivial step. And how do we know if we should take account of all the data that we've access to, or whether at some point we've got enough data and we don't need to move to increasingly complex discussions of how we do that? Jim fairly firmly told us that it was important that we had to assess the performance of the new data sets versus some index data set or a traditional model or whatever. But on the other hand, even if the new models work, we had a significant problem in persuading people to adopt them, was the take-home message I got from one of the take-home message I got from Jim Stevens. And in addition, we had to be skeptical as to how easy it would be to move an organization or the world or our peers or whatever it was to accepting a new paradigm or a new model in the session 1 definition of model, not the session 2, within the organization. How do we move an organization to accept a new way of thinking about toxicity, or a new way of identifying toxicity, or a new way of identifying efficacy when people are awfully comfortable with the methods they've used before? As he said, it was pretty convincing if you hadn't started yet. It was not so convincing if you'd already got data that you were wedded to. So I think we heard the challenge of using the full, using access to all the data we could get our hands on, but on the other hand, heard the limitations of using all that data, some of which were as traditional as people in the 1800s would have told us. People are immovable and well wedded to what they believe, rather than moving to new data sets, and that's a traditional challenge, I guess.
Thanks. Well, I'm the last one on the program, standing between you and the evening activities, and so my comments are going to actually be quite brief, and mostly because session 2 was the bulk of the day, and I don't plan to summarize every single presentation. It would be near impossible. But what I heard over the course of the day is confirmed what I've thought for quite a while, which is that we live in an era of incredible innovation. And we live in an era of incredible innovation in a whole variety of settings. And there are certainly a variety of settings which were presented over the course of the day in which information and the integration of information is becoming increasingly the norm, and I think is increasingly making a significant difference in terms of the way that we're able to approach a whole variety of problems. We heard about the way information and the integration of information is being used in the biomarker space, it's being used in clinical trial design, and is being used in pharmacovigilance. And the tools are becoming more sophisticated, the data opportunities are becoming richer and becoming significantly more abundant, and the analytic and modeling tools that are available are becoming better and better. So as there are many that believe that more is not necessarily better, but I think that there were certainly many examples presented over the course of the day in which more is certainly better, and I think that will very much serve the regulatory science community, and certainly will serve well the regulators that have to ultimately make decisions about what to do about various medical products. So I will end by coming back to a statement that I made earlier today, which is from the standpoint of the regulator in the room, that increasingly we're looking at science on the edge of their comfort zone. And certainly, I've heard a number of presentations about science that would generally be considered outside of our comfort zone. But increasingly, I think the information that I heard today certainly tells us that we can use many of the tools that were presented to be able to make regulatory decisions. And that, I think, is ultimately to the benefit of the entire enterprise, whether it's those that are doing basic research, those that are doing translational research, those that are engaged in regulatory science, and ultimately the ones that we do all of this for, which is the patients that need many of these products. And so I think what I've heard certainly bodes well for the future in terms of the way that we will be able to use some of the things that we heard today to make the best decisions for patients who need these products. And so with that, I think we will end the day, and tomorrow we will come back and hear about some of the very important infrastructure needs to be able to do many of the things that were discussed over the course of the day. You just don't snap your fingers and make all of this happen. It takes people, it takes resources, it takes ideas, and that's what we'll be discussing tomorrow. So I look forward to that discussion in the morning. Thanks, everybody.